Dus Zen zou ook meedoen? Of was het alleen
Zeker, bedoelde ik.
Oh, ze kan je.
Okay. Good morning, everybody. So welcome to this final day of the Handling Lifelines data on the UMCG High Performance Cluster course. So today the topic will be about the genetic data of lifelines. And the first speaker will be Harold Sneeder, who's professor in the Department of Epidemiology in the unit of Genetic Epidemiology. And the person who is one of the initiators of the UGLI consortium. So with that, I would like to give the floor to you, Harold, and we are eager to hear what you have to tell about the Lifelines genetic data.
Thank you very much, Ilya. I'll be happy to speak at this great course. Let me share my screen. We tested it, so it should work. Okay, now I see my PowerPoint's no longer there, so I have to first find it. Okay, so I hope you can all see my presentation.
Yep, I can see it.
Okay, I'll be talking about the Lifelines genotype data in this first 20-25 minutes or so, which consists of two types of chips or arrays, the CytoChip or CytoSNP array, and the global screening array that has been used in the UGLI data. So first, a little bit of background. What is Lifelines? Well, Lifelines, as most of you probably know, is a population-based cohort and biobank with over 165,000 participants. And we intend to follow these people for at least 30 years. It's a 3-generation design, so family members have been included. Really what Lifelines is a research infrastructure that is available to researchers not only within the Netherlands or Europe, but really worldwide. And the aim of Lifelines could be described as building a research infrastructure for population health and precision medicine. So what has been collected in terms of data and samples? Well, as you have heard, there's a lot of phenotype data. This is just the phenotype data of the baseline. So we have a lot of physical measurements. We have a whole range of biomarkers, laboratory assessments from blood and urine, questionnaire data, and there is record linkage data that can be also very interesting for answering different types of questions, for example, on exposures to air pollution. So we're really talking about big data for health, because everyone realizes that the future lies in personalized healthcare and precision medicine. So for that, it's needed to shift from a group to an individual level. And big data is needed to improve patient care and public health, we think. And what is essential there is that also in Lifelines, but also other biobanks, there's many participants, there's broad data collection, there's linkage with other data sources, and of course there's biomaterials such as DNA, which is what we are talking about today. So Lifelines got some funding from the NWO quite some time ago. I think this was in 2008, to build a genomics infrastructure for a population-based cohort of 165,000 individuals. That was the title of this NVO groot application, very large infrastructure subsidy. So this was realized around 2012 as based on this funding from NVO. So we built a dynamic ICT and data management infrastructure, including an online catalog. And you've probably heard about this, or you will hear about this, where you can just browse the data, see what's available in LifeLines. DNA was isolated and stored of most of the LifeLines participants, about 93% of the total biobank. And genotyping at that stage of about 13,000 DNA samples of the CytoSNP SNP array was done, and later an additional 1,500 samples from LifeLines deep cohort, as a sub-cohort, were added. So for a total sample size at that time of about 15,000 individuals. So what did LifeLines do with this genotype, this genome-wide association data? Well, we started to participate in international consortia doing GWAS studies. And the principle of a GWAS study is shown here, where you have a large patient sample with a certain disease and a large control sample. You can genotype these on a genome-wide association chip, then they will contain at least 500,000 SNPs. And then you can compare the allele frequencies of those SNPs between the controls, the healthy controls, and the patients. And you get what's called a Manhattan plot, where each dot represents a p-value of a statistical test, comparing the genotypes of those two groups. And as you can see here, there is a peak, and the y-axis is the p-value. So higher values mean lower p-values, so more significant. So this is a really very significant signal. And then typically what you do is you contact your colleagues all around the world with different studies, and you ask them, can you replicate our signal? So you get independent replication of top results, and then in the end you get your odds ratio, the effect size of a particular signal or SNP, or many SNPs that you are finding. So that's the principle of these genome-wide association studies. There's a GWAS catalog that you can look at, and this is the code book of such a this GWAS catalog, and each dot represents a different phenotype that has been looked at. And that GWAS catalog gives you an overview of all the things that have been found since the first successful studies that were published around 2005, 2007. So what did LifeLines contribute to all these GWAS studies? Well, we contributed a lot with our 13,000 CytoSNP data. A large GWAS study on blood pressure, for example, obesity, but also other anthropometric traits, heart, like electrocardiogram traits, lung function traits, liver enzymes, for example, but even a GWAS of educational attainment, which at first sight seems strange that there is a genetic component to that, but there certainly was. We were successful in showing that. And not only did we participate in these consortia, a number of consortia or studies were also led from the UMCG. And the first example was the very successful GWAS study of Dupuytren's disease. It was just published in New England Journal of Medicine in 2011. Another one was a publication in Nature, led by Pim van Harst from cardiology, and colleagues looking at loci for influencing human red blood cells. A study we were involved in was looking at number of children and age at first birth, so really human reproductive behavior, which was published in Nature Genetics. And then a big study of our own group, led by Ilya as the first author, on heart rate variability. So there were, over the years, many LifeLines publications, many genetic publications, particularly more than 100 now, and most of those were published in top journals, such as Nature, Science, Nature Genetics, etc. And what we decided to do is use the LifeLines cohort study as a group author in publications to improve visibility of LifeLines. And also we published what we called a genetic position paper in the European Journal of Human Genetics. So again, Ilya and Peter and our group were heavily involved in this, together with the other main PIs of the UMCG involved in genetic studies at that time. And this led to a nice paper in this European Journal of Genetics, focused really a showcase paper focusing on all the quantitative traits that we had measured and available in LifeLines from 6 disease areas. And they are listed here, and then showing the quality of the genetic data in relating genetic risk scores to those particular outcomes. So what about the UMCG Genetics LifeLines Initiative, also known as the UGLI Consortium? Well, we were involved in collaborations with a group from Peter Visser in Australia, and also they were using some of the LifeLines data. And this is really a very pioneering group, publishing lots of different methods, but also very innovative ideas in what to do next. And this paper addressed the question how to design the next experiments. You know, should we focus on sequencing maybe, because that became more and more affordable, or should we still do focus on GWAS studies? And there were a number of developments at that stage around 2014, 2015. And one of them was that there were larger imputation reference panels that were greatly improving imputation accuracy for rare variants, even for GWAS chips, for GWAS arrays. So for further discovery of genes associated with complex traits, it was concluded a study with SNP arrays followed by imputation would be much more cost effective than whole genome sequencing at that current price, because sequencing was still quite expensive, and SNP arrays became actually quite cheap at that point in time. And so it was easier to achieve large numbers. So really what we decided to do when Andre Uiterlinde from the Erasmus MC started organizing to have your cohort genotyped using the Illumina global screening array, we jumped on the bandwagon, and we joined Andre Uiterlinde's project, where he negotiated a good price with Illumina for having all these cohorts genotyped. His task was to get at least 500,000 samples, and then he would get a major discount. So we participated also with LifeLines in this. Then at that stage, Siska Wijninga was still head of genetics. So we came together and we said we need to participate in this project, have eventually all lifelines genotyped. That would be our goal. And we founded the UGLI Consortium. So what we did was we simply contacted all our colleagues here at the UMCG and the RUG, and we brought together financial commitments of more than 25 PIs. They were interested in genetic research in lifelines. And by October 2016, we had collected commitments for over 1.6 million by these researchers and the board of directors of the UMCG. And then after negotiating with Lifelines in September 2017, we signed an agreement. We agreed that all these participants that were investing in genotyping for Lifelines would get freely accessible data for at least 3 years. Lifelines agreed to provide one UGLI phenotype data set that all these researchers could work on, and short research plans for each project were needed. So we created a dedicated short UGLI proposal form, and some of you have already used this and submitted this and got approved to actually work with this data. And these proposals are evaluated by both Lifelines and the UGLI steering committee that currently consists of myself, Lude Franke, Patrick Delen, Jan Albert Kuivenhoven, and Peter Visser. On top of that, many clinical UMCG cohorts, but also clinical cohorts throughout the Netherlands have been genotyped using the same genome-wide Illumina GSA chip. Just some examples here: the GLGS for glaucoma, Peyronie Dupuytren for plastic surgery. Gastroenterology had IBD patients genotyped, and neurology Parkinson's patients. And the UGLI data gave them the opportunity to use them as control groups to do a case control GWAS. And several of these case control GWASs have indeed now been done with the current data. So the ultimate goal was to genotype entire lifelines. We already had 15,000 cytochip data, and then in January 2020, an additional 36,000 GSA samples were released. So people have been working on that data ever since. Currently, genotyping of an additional 30,000 samples with yet another array, the Axiom chip from the competitor of Illumina, namely Thermo Fisher, is ongoing. They gave us a good deal, so we decided to sign a contract with them. They delivered 30,000 samples, and that genotyping is ongoing. We currently are at, I think, 13,000 in spite of all the COVID restrictions. And yet another 30,000 samples will be funded from Australia, from money from a grant from Peter Visser. So one of the things we also did together with Lifelines is to share one UGLI computer environment, or to create this computer environment on the high performance cluster computer, together with Morris Sretz, his group, that are also involved in teaching and with the genetic department. And we developed this course to teach you how to work in this environment. So with that, I would like to thank you. This was just a little bit of background on, on the history of the project, how we came at this stage where you have one nice computer environment, one large data set that everyone can work on using harmonized phenotype definitions, for example. And the floor is open for questions. Ilya, can I hand over back to you?
Sure. So any questions to Harold? Please take the mic or in the chat. Yes.
Hi, just a kind of general question. I'm just wondering if I didn't miss the talk. Is there also a plan to have exome sequencing, or does all the genetic or the UGLI initiative includes only whole genome sequencing?
That's a good question, Bale. There is some data available, I think, through Ecology on the exome chip, but that's just a very small sample. That's really not involving the UGLI initiative. So really what we're focusing on is GWAS chips. And there has also been a project called GoNL. And this was done in 2013-14, where 4 cohorts in the Netherlands, including the Rotterdam study and also the Netherlands Twin Registry, but also LifeLines, were involved in whole genome sequencing.
Okay.
But that's a fairly limited sample size, I think about 600 or so. And I think 120, sorry?
150 trios from LifeLines.
150 trios from LifeLines, yeah.
250.
250, okay. Yeah, so the parents plus 1 child were whole genome sequenced. And this data we are still using actually for quality control purposes of our GWASs. So we compare obviously the GWAS genotyping to the whole genome sequencing of these 250 trios. That has been used several times for the last chip and also for the new chip.
Okay.
And these GoNL data is also available, but not via LifeLines. That's via GoNL itself.
I see.
Yeah, it's publicly available, and it's also been added to the high performance. No, not the Michigan server. So the Haplotype Reference Consortium, HRC. So GoNL is part of the reference data set that is being used for imputation. So when we impute our data against the reference data at the Michigan server on about 30,000 whole genome sequence individuals, the GoNL data is part of that.
Yeah. Okay, thanks.
Other questions? No, I don't see or hear any other questions. So thank you.
I have a question for the participants. How many of you will be using the genetic data? Just put your answer in the chat. Tai Chi will use the genetic data.
Sakai, Doreen.
Okay. Doreen, probably. Iran as well, yeah.
So Tai Chi, what is IADB? Pharmacy data, okay. Interesting, so pharmacogenetics.
Yeah, that's the pharmacy data indeed. I'm familiar with that, so yeah. And that I think Eelco Hak has been involved in linking. So that's one of the data linkage projects in linking that data to LifeLines. So most of you will be working on the genetic data, so that's great. What about Joost? I didn't see a response from him in the chat. He may be making some coffee. Zekai, you have a question. How to impute? Ilya, is that addressed in the course, the imputation, how that works?
No, the imputation is not addressed in the course, but I can say something about it. So the imputation, so the first step of the data is when you get them from the lab is to perform quality control. So that has been done extensively in a working group in which I was also involved. But this was for the UGLI data was performed by the genetics people from the genetics department. So then when the data was quality controlled, so the clean data, they have been well, actually they have been uploaded to a server from the Zanger imputation, from the Zanger Institute. They have an online server where you can upload your data in a particular format, and when you upload it there, then in a few days you get it back from them, and that's actually it nowadays. So you don't have to do the whole imputation yourself, but it's just uploading your data and downloading it again after a few days.
So this used to be much more difficult, right? When we had to impute our own data.
Yeah, in old days we had to do it ourselves using impute. We used impute for several other cohorts. It's much easier nowadays.
And the general principle of imputation is, for those that don't, is that the genomic structure or the structure of the genome is such that many variants are highly correlated. So if we have reference data, we can predict what the SNPs that have not been genotyped will be, and that's the principle of imputation. So those predictions of non-genotyped SNPs are actually filled in your data set, and you can then use those in your analysis. So the standard really these days is to use imputed data in all your analysis. And typically the imputation quality is really good because the high quality of the reference data and also of the array data, the genotype data.
Yeah, so for instance, for the UGLI data, that we had data on, I think about 550,000 or 600,000 SNPs that were genotyped and passed the quality control. And then with imputation, you extend your data, well, with the haplotype reference consortium panel to about 40 million SNPs. But of these 14 million SNPs, many are very rare variants that are, well, because it's a global reference data set, many of those are not present in the European population. So when you filter out those variants that are non-polymorphic, so monomorphic, then you have about, well, 10 million SNPs, which is still a very, very large number.
Yeah, these are really big data that you have to work with.
Yeah.
But as soon as you calculate, for example, risk scores, polygenic risk scores, then it reduces to a very manageable number of new variables. But for GWAS, for example, we use all these different SNPs. We do regression analysis for every SNP separately. So we do a lot of tests that we have to correct for.
Okay, other questions?
Yeah, other questions? If not, I hand over to you, Ilya, for continuation of the course.
Yeah,
and
thanks.
I wish everyone good luck.
Thank you, Harold, for this nice overview of the LifeLines genetic data.
My pleasure. Okay, bye-bye.
Bye. I will go over to the next presentation. Which is about how to handle the LifeLines genotype data on the UMCG high performance cluster. So in this presentation, I'll be using the genotype data on the cluster and give you some tools how to handle these data, how to view them, and how to manipulate or deal with the data. And then the final item of this morning is how to add the geno ID to the phenotypic files, because in the genotype files another identifier for the samples is used then in the phenotype file. So where can you find this linkage file? So first about the genotype data. Well, as Harold said, there are two chips. The earliest one was the CytoSNP chip that was used, and the data were released in different releases from 2011 to 2015. But then the UGLI consortium was initiated, and their data has been genotyped with the global screening array chip. And here in those genotypes, you can find the cleaned data, the genotype data. So that's the data on these 550,000 or 600,000 SNPs. And here is the imputed data. Well, the CytoSNP is, as I said, an older chip, and for this SNP, the genotype data was only available for about 250,000 SNPs. So you can imagine that the imputation quality of this SNP is a bit worse than this for the GSA chip. So when you have to use this GSA chip, but of course you can also use both data sets for analysis and combine them. I would not combine them, the genotype data itself, and then do an analysis on the combined data set. But if you would like to use both cohorts of both data sets, then my advice is to do an analysis on the GSA chip separately from those on the Cyto chip. So for the UGLI data or the GSA chip data, well, there is raw data, which you can have a look at, but I would not advise you to it because, for analysis, it's of no use. You can use the genotype data that has been cleaned. In this folder, GSA genotypes, you will also find the scripts and the logs that have been used or have been the result of the quality control. And here you will also find the principal components to adjust for population stratification. So there are 20 principal components, and the advice is to use at least 5 of them in your analysis. Then these data have also been imputed against the haplotype reference consortium reference panel. So the size of the data was increased to about 40 million SNPs, as I said. And the haplotype reference consortium will give you back VCF files. I'll come variant calling format data, and I will show you what this looks like later. And within the Department of Genetics, they also converted these to other formats, the BGen, that's binary Oxford format, and GDS format, which is a format that can be used by a software called SAGE. And this SAGE software can deal with the relationship between the individuals. because LifeLines is a 3-generational design, and for the ugly design, we selected the largest families. So there is many relationships within the ugly data. So you have to correct for this genetic relations between the individuals, because these data are not independent of each other. So the individuals are not independent. So you cannot use just regular, well, let's say linear regression. But I will come to that in the afternoon. Then for the CytoSNP data, not everybody may have access to these data, because you have to specifically, well, also for the ugly data, you have to request those data. But most of you may have asked access to the ugly data. So then it's not automatically that you also get access to the CytoSNP data. And on the cluster, if you can, if you did request them, you have, well, there's also the raw data, but well, it's of no use, and also you don't have permission to look at it. Well, at least I don't have permission to look at it. The genotype data are there, so for the 250,000 SNPs that have been cleaned. Unfortunately, LifeLines did not put the PCs on there, although I have recently recomputed them and asked them to put them up there. But they have not yet done so. But they are available. So if you would like to have them, if you want to do an analysis on the CytoSNP, just ask the LifeLines data managers for those data. These data have been imputed using the 1000 Genomes reference panels. That's a predecessor of the Haplotype Reference Consortium data, which has well, a little less individuals in their reference panels, 65,000 compared to 100,000, but still the quality is well, as good, almost as good as of the Haplotype Reference Consortium. Well, these data are for about 15,500 individuals. There are, well, the first 13,500 were to be independent, but then with the LifeLines Deep was related individuals were added. So also these data cannot be analyzed just by, well, let's say linear regression. So therefore also these, well, the first release of the CytoSNP data was only in this gen format, which can be handled with, for instance, Plink or a SNP test. But as I said, it also contains related data. So also for these for this data set, SAGE is the designed analysis platform and analysis software. So therefore, the genetics department also converted these CytoSNP data to the SAGE GDS format and also to VCF formats. Then where can you find well, the imputed data? So that's the most well, important or data on the cluster. These imputed data, they contain the genetic data of genotype data, but it is extended to about 40 million SNPs, as I said before, using the Haplotype Reference Consortium. Well, these data are in the GSA imputed directory, and then in the subfolders V1 VCF_ files. And in there, you have all, for each of the chromosomes, you have a separate file. So from 1 to and so on till 22. And also the X chromosome is there, which is not available for the CytoSNP data. But you see here, if you with LS minus LH, you also get the sizes of these data, and you see that these files are huge. Oh, well, the smallest one is 2.6 gigabytes. So it's not well, these files are not easy to handle. In total, there is slightly less than 200 gigabytes of data. So how do you handle these big files? Well, do not unzip them, because then it will take up a lot of your time and of disk space. Also, do not edit them with Nano or Mobile Xterm editor, because they will run out of memory. So the advice is to use, or the way to do it is to use the zcat command. So this is a command to unzip the file. But if you put here this vertical slash and you can put other commands after it. So if you do it like this, then it will unzip the file in memory and then do additional commands, executions on this file. So as I said, you can concatenate commands with this vertical slash, and then it will perform actions in the memory and only print the results of the last command. So for instance, if you do it like this, zcat of a gzip file, then say cut minus f1 to 10. This means print the first 10 columns, well, print in memory. And then do head, then it will print the first 10 lines. So the result of this will be the first 10 lines and the first 10 columns. So if you do it for instance, this chromosome 22 ugly imputed data, then you will get this result. So it will show you the header of the file, the first 10 lines. Well, there were no 10 columns in here, just one. So it will not. It will only show you, well, in this case, the complete headers. Well, with head, you can, as I said, you can just see the first 10 lines. You can adapt it to say minus n100 to view the first 100 lines, but better is to use less command and to scroll through a file, and then I'll use this option minus capital S to truncate the lines, because otherwise you will get more than these all these 36,000 columns on screen. So that's not very convenient. So if you do it like this with this minus of less minus S, then you get a result like this, which tells you that well, and I have scrolled down within this file to get to the genetic data, and you see something. You see here the genetic data. That's after after 100 lines or so, you get to the data. So the header of the file is about 100 lines, and then finally you get to the data. So here you get the genetic data telling you on which chromosome is, for each line is a SNP. So this is a particular location on chromosome 22 in this case. On this is the base pair position. Here the ID is the SNP ID, so that will be well, could be the RS number if a SNP if such as SNP identifier is known, then there would be an RS number in here. Then it will give you the reference allele and the alternative allele. Then we'll give you some quality filter and info, but this might not be filled in. I think so here. So the quality, this dot is for quality, so quality is not filled in this file. Filter is set to pass here, so it passed the imputation quality filters. But I believe everything is here a pass, and it's for analysis. It's not necessary to check for this. You can just analyze everything and do a quality control on the results afterwards with, for instance, GWAS inspector. More on this is discussed in the advanced genetic epidemiology course. Then it will give you some information on the SNP. So what's the allele frequency in the reference panel? What's the total number of alleles counted? So that's this is 36,000 plus times 2 for the number, because everybody has 2 alleles. Then AC is the count of the alternative allele. So here it says 0 C alleles have been counted within these 36,000 individuals. The info is the imputation quality, so that's here 10 to the minus 9. So the imputation quality for this particular SNP is very low, but this is well, the SNPs are sorted in order along the chromosomes, so sorted on base pair position. So, and it's usual the case that SNPs in the beginning of a chromosome and SNPs in the end of the chromosome and SNPs around the centromeres of the chromosomes their imputation quality is lower, because it depends on the linkage disequilibrium structure with, well, the first SNP that is present that has been genotyped for this chromosome. So if that's far away from this particular SNP, then the LD is low, and then the imputation quality is also low. So the stronger the linkage disequilibrium, the higher the imputation quality or info score. Then this will tell, this is the format column. This will tell you what kind of genotype data is there, where GT stands for genotype. That means the best, the most likely genotype. DS is for dosage, so that's counting how many of alternative alleles this individual has, taken into account the imputation uncertainty. And GP, that's for genotype probabilities. So that gives you the probabilities of the genotypes after imputation. So with imputation, you get the probabilities for the, well, in this case, TT, TC, and CC. What's the likelihood that this particular genotype is in that position? So this is referring to what the data that are in the next columns. So here the GT, that's referring to this 0/0, meaning that this individual has not a C allele in the second position, in the first chromosome, and not a C allele in the second on the second chromosome, meaning that this individual is homozygous for the T allele. And this is the dosage, 0.1, and that's calculated from these 3 probabilities. So the probability that this individual is a TT is 90%. The probability that this individual is a heterozygote, TC, is 9.5%. And probability that this individual is a CC is only 0.25%. And the dosage, this 0.1, is then calculated as 1 times this plus 2 times this probability. So that's where this dosage comes from. When you analyze the data, you can either, most of the times, this DS, the dosage, will be used in the analysis to correlate this dosage with your outcome. Any questions on this? No. Thanks. Then I'll continue. So then, but this is just the start of the chromosome. There are many SNPs within this data file, and you can scroll through it. And also within these, if you have this less command, you can search in your output with the backslash and then /, and then put your search term. But you can also use other tools, and one of the things is the grep tool. which you can use if you put in zcat and then the file and then put grep. Then it will give you, then it will search for the string, well, in this case, 1, 2, 3, 4, 5, and it will print the lines that contain this string. If this is, you can specify if this is a whole word. So it might be that one of the genotype probability is 0.12345, but if you are searching for this in a position, for instance, then this is a whole word. You can add this -w option to search only for whole words. Or if you, well, this would be a text string, you can use -i to ignore the case, so to search both for capitals and small letters. Well, if you do it like this, it will print the output on screen, but you can also redirect it to a file, and then it will save it the result to this 12345.vcf file. Alternatively, if you want to search for this in a particular column, so let's say in column 2, which is for the position, then you can do it with an awk command, saying for what if position, so if column 2 is equal to 1, 2, 3, 4, 5, then print the outcome. Then it will print all the lines that have 1, 2, 3, 4, 5 in column 2, and this is then this means that the whole word has to be 1, 2, 3, 4, 5. And note here, I use here this backslash. So that's, if you use backslashes, then the command will continue if on the next line, but don't put a space after it, because then it will not recognize it as well, end of line. If you do it so without the space after this backslash, then your cursor in the console will change to this this arrow sign, and you can continue with your command. So this is convenient if you have well, many commands that you want, many commands that you concatenate. Then it's for an easy layout of your command in your script. It's easy. Convenient to use this bash. You will see it in well, in the exercises. So as I said, the cursor was changed, is then changed to this well, this arrow sign. If you do not want, then the bash will still thinks that you're working on a command. If you want to quit it, press Ctrl+C, and then you get to the normal cursor, the $sign. This awk command can be extended. Awk is well, very nice way to put more complex things in here within. So here, if for instance, this tells you to also not only print the columns of the lines that contain 1, 2, 3, 4, 5 in column 2, but this will also print the first 10 lines. So for a VCF file, it's convenient. Well, it's convenient. You have to retain the header. So in this case, if your header would be 10 lines before the genotype data starts, then use this to print the header, and then only those genotypes that have, well, 1, 2, 3, 4, 5 that are in the second column. And what more? You can change this awk command, for instance, like this, saying if for the header lines I want to print the complete lines, but for the other lines I want to that contain 1, 2, 3, 4, 5 in column 2, I only want to print the columns 1, 2, 3, 1. So that's I don't have any idea why you would do that in this case, but this is more an example of the awk commands. Or alternatively, if you do it like this, if in lines in the first line, no, what in the header, no, if the lines are above 109, so from lines 110 and on, I want to change the ID in position 3 to a chromosome. What's the semicolon? No, it's not semicolon. What's the word? Well, this, and then the position. So I want to change this ID. Then you can say, okay, I only want to change the ID. So that's column 3 for lines 110 and on. But no, I mean this column. Thanks. Yeah, so then it will change column 3 for only for the genetic data, so not for the header. And you see it like here. So this is the would then read the result. So here you concatenated 22 and the position 160. What is it? No, 60 million and so on, and separated by a column. So you can do a lot of things with awk, and also these within these curly brackets, you can put in a lot of things. How do you want to manipulate the file? So it's well, more or less a scripting language of its own. And here's a website where you can find more about awk. Also, if you want to have things in mind, you can Google and say what you want to do with awk, and you can. There are fora who can help you, so you can try out what you want. You can also put loops, for instance, in awk, saying I want to only to print every odd column or every third column. So it's a very nice tool, this awk. At least that's what I think, but also pretty complex. Well, with all these Unix tools, you can you can do nice things with these VCF tools. For instance, extract data. But there is also a dedicated tool called VCF tools. Earlier there was VCF tools, but now there's a newer tool since a couple of years that's called BCF tools, which is can do even more things and is more efficient. Information on VCF tools you can find on this website, and as I said, it's an extensive tool for manipulating VCF files. You can do a lot of things with these VCF files. Conveniently, this module VCF tools is available on Gearshift, so you can do with module load VCF tools. You can load the module into your workspace. And to get help on VCF tools, you can type VCF tools. Then it will give you a list of commands, things that you can do with VCF tools. And if you then do VCF tool commands, then it will give you a list of options for this particular command. And the commands that are convenient, that are there for VCF tools are these: annotate to edit VCF files and add or remove annotations, concat to concatenate files from the same set of samples, convert to other formats, for instance to Plink format, filter. You may want to filter on allele frequency, imputation quality, or this pass filter column. Merge. You can files for non-overlapping samples with same set of SNPs. You can sort the files. You can produce statistics on the files. And you can use view. That's convenient to make a subset of the file. So with this view, you can make as I said, you can extract a subset of SNPs. For instance, if you have my BCF tools view and this minus small t, you can give in a range, an interval on chromosome 22, let's say from position 100,000 to position 200,000. You can save this file with the -O option. You give in the name for the file, so snip file vcf.gz, and with this O, capital OZ, you tell it to zip the file, the results file. And here you tell on what's the input file. Alternatively, so here with this -t, small t, you can give in an interval. Alternatively, with capital T, you can give in a list of SNPs. So that's more convenient. Most of the times, you would rather, well, no, you could have an interval if you are interested in a specific gene. Or if you're interested in specific SNPs, for instance, from GWAS hits, you can have a list of SNPs that you want to extract from the file. Then you can give in with this capital T, you can give in a list of SNPs, and this SNP file then is a text file with a tab delimited text file with chromosome and position in it. So then it will, then the results in this result file, it will give you the SNPs that you request, and that can be found within this, this input file. Then another convenient tool is to merge data. So you can merge data from multiple VCFs. With concat, you have the concat and the merge commands. With merge, you merge files with same samples but different SNPs. So for instance, you can merge the samples from ugly chromosome 22 and chromosome 21 together. With this is with the concat. With the merge command, you can merge files for same SNPs but different samples. So for instance, if you want to merge the data from the ugly chip with those from the CytoSNP chip for chromosome 22, then you could do it like this. Then the nice thing is to annotate, because as you saw before in the ID position, there is often just a dot. So that's not very convenient. So you can annotate, you can fill in the SNP ID. Well, for instance, with this, if you put in here annotate and then -- set ID, then and fill in that the chromosome underscore position underscore reference allele underscore alternative allele, or first alternative. Sometimes if you have multiple multi-allelic alleles, then there will be more than one alternative allele. So therefore, first alternative allele. Then it will fill in an ID with chromosome underscore position underscore reference allele underscore alternative allele in this third column of the VCF file. Or if you want to have RS IDs in there, you can use an external file. This is a file that you can download from the internet, this dbSNP. This contains the positions and RS numbers for every SNP, for many SNPs, not every SNP, but for all that are there in the dbSNP database. And then it will extract the RS numbers from this file and put them into your file. So this is also very convenient. Any questions on this? No, I don't see. Put them in the chat or raise your hand or grab the microphone. So you will practice with BCF tools and and with these Unix tools to get an idea of the data that's there on the UMCG high performance cluster. So then next thing is that the IDs, the individual IDs, are not the same. In your phenotypic data, you had this project pseudo ID, which is kind of these long barcode-like IDs. While in the genotype data, you have, well, for the GSA, UGLI1, UGLI2, and so on. And for the CytoSNP data, there are identifiers like LL, 016774, and so on. So you have to, you have to know which individual, which identifier is the same. And these data are in the GSA link or CytoSNP linkage files. So, for instance, for the GSA linkage files, there is this pseudo GSA linkage file project pseudo ID file, which tells you that this project pseudo ID in the phenotype file is the same as this UGLI ID in the genotyping files. And there is also a third name saying that's the genotyping name for the lab. So that's the individual ID. This is the plate ID on which this individual was genotyped, and the well in the plate in which its DNA was put for analysis. So you can merge your genotype ID to your phenotype ID using this join command, saying that you want to join the phenotype ID phenotype file with the key in the first position. The linkage file, the key is also in the first position, the first column. If you do that, then well, in this case, I got an error because it says that the files is not sorted. So an important thing for this join command is to sort the files on the key. Well, you could do that. You can do that in R. You can also do it here with this Unix command, but then you have to put it in here. You have to put in that you want to use the file phenotype file that has been sorted on the first position. So you put here this smaller than sign. No, don't put a space in here. Just directly after this smaller than sign, put the round brackets. Say sort pheno file and sort it on the first column, on the first key. And then close the bracket, and the same for the linkage file. So if you do it like that, then it will sort your data on this and use the sorted data for joining. And if you do it like that, then you get this result, which looks okay. Any questions on this? No. Okay. Next slide. Why don't you want to do that? Oh, there. Okay, then I'll explain for the exercises. So first thing is to go to Nestor and upload the scripts file that's there for the exercises of today. The next exercises will be on viewing how the UGLI GSA data looks like to get a feel of the data, extracting a subset from these data, and adding these UGLI IDs to the phenotype file. So that will be that will be the topics of the exercises of this morning. So go for to the Nestor page to download, well, the exercises, the answers. Well, there's actually no data. Only, well, there is one data set that's called exclude. Well, you have to create it, but well, it's one of the exercises is to create this data set in the afternoon, but that will take too long. So therefore we also provide it to you. But there are scripts which you also have to download. So exercises, the answers, and the scripts, and one file that's needed in the afternoon. That's exclude GSA relatives. Okay, so in case you have a question during the practicals, you can post your question in the chat, or you can put it in a general chat or to me in particular. And possible we can share screens if more help is needed. And then the next lecture will already be at 1:30. Okay, so with that, please go ahead with the exercises, and I'll be here in case of questions. Good luck.